This is an OCR A-Level Biology video on basically Unit 1. As you can see, this video we're looking at basically accuracy, precision, validity, reliability, how to answer those sorts of questions on experiments. So over here we've got limitations of the experiment, and this is what I'm looking at later. So how you might get a question saying what are the limitations of the experiment, and then this is the sort of things you should be speaking about. I made a little analogy there, and I'll speak to you about that later. Looking at the design of an experiment, and sometimes you get a question pretty much asking you to design an experiment, and I'll just explain what you really need to be speaking about there. And also we're going to look at accuracy, precision, validity, reliability, what these terms mean and what sort of things we can do to improve them. And we're going to start off the video by looking at variables, where they belong in the graph, and just some general tips and tricks. But once again, we're the first cohort to ever do this exam. There's not much past paper material. Often some of my points I make are made from other exam boards, other mark schemes. I'm not going to guarantee that everything I say is going to be in a mark scheme, but I think it's going to give a good, probably if you can get a 6-mark question, if you write the sort of things I've told you, you get 5, 4, hopefully 6, but no guarantee. Anyway, let's start. I'm not going to write out as we go along this time. I'm just going to speak you through each thing because I've only just made this on the spot. I saw a comment. Thanks for the comment, by the way. It's actually going to help me as well by making this video, because it is somewhere where I lose a lot of marks in papers, something that you never really think of revising. You just want to revise the content. Anyway, enough talking, let's get to it. Variables. So you've got these 3 variables. Dependent is the one you measure, independent is the one you change, and control are the things you keep the same. But you should also have a control, so a negative control, and that is usually distilled water. So you might be measuring like the effect of substrate concentration on enzymes, which we put here. So you'll be using a different concentration of substrates, and to prove that it's not water having this, water having this effect, you just do the experiment without any substrate, just adding distilled water, and you'll get a negative result where the rate of reaction doesn't change. So that is always, this control is always going to be different in different experiments. But if you don't have a clue, my advice would be stick distilled water down. It can usually be a handy thing to put down. It's usually in most mark schemes as a control. But for stuff like measuring light intensity, measuring photosynthesis, you could just use a dark room, so a plant with no light affected on it, and just to show that light is definitely having this effect. Okay, so they're your 3 variables. Dependent, you always put on the y-axis. Okay, so that's the one you measure, so like rate of reaction, and the independent always goes next axis. So this question says effect of temperature on the rate of reaction. So if we're looking at the effect of temperature, therefore temperature is the thing we're changing. It's our independent variable. It's going to be on our x-axis, so at the bottom, and we're going to have rate of reaction on the side. When you have to put your own graph, always put your units, so degrees, rate of reaction. It depends what you're measuring. It could be oxygen uptake. I've just put arbitrary units on the side here, but remember, always put your units when you're writing a graph. Always take your time, plot the right stuff, and make sure your independent is on your x-axis. That's the thing you're measuring, and it's the same here. So if we did effective substrate concentration, your substrate concentration will be at the bottom, rate of reaction on the side. They're just the 2 things. I've done an enzymes video pretty recently, and that's the thing that was in my head. One other point here, it's just a general point. This thing will get you a lot of marks, reliability and validity questions, if you state repeat the experiment you will not get the mark. If you state repeat the experiment a minimum of 3 times, you will get the mark. That is, and you might not believe me, so I've literally put in a picture from a practice paper based on our specification. So a specimen paper we got, this only teachers has access to it, recently did it, and look what I put. So it's asking you about validity, 3 improvements you can make to the design of the experiment, and I put repeats. The student only tested 4 individual plants. The experiment should be repeated with different individuals. That was not worthy of a mark. If I put repeat a minimum of 3 times, I would have got the mark. Okay, so always state how many times you're repeating it, and it's got to be a minimum of 3. Okay, you could say 10, but just I'd advise you just say repeat a minimum of 3 times. Okay, so that is some general tips. Now let's look at accuracy, precision, validity, and reliability. So accuracy is when accurate results are really close to the true answer. Human interpretation of a measurement, such as determining a colour change, can reduce your accuracy of results. So as it's defined here, the degree to which measurement represents the true value of something. Simply put, how close a measurement is to the true value. To make sure you've got accurate results, I've put this thing here. You want to be using quantitative measurements rather than qualitative. And if you look back to the biological molecules test, when you're using Benedict solution to measure the amount of reducing sugars, so you can get that gradient of colours. So you can get the green, yellow, red, orange, red. Okay, and they all tell you different concentrations. If you're using the human eye, it's really subjective. It's a matter of opinion how different the colour is, and I think everyone knows that people have different opinions on colour and how dark or how light something is. I know I've had plenty of arguments whether something's orange or red, and obviously the object is definitely the same, but people just view it differently. So to avoid that sort of human error, that subjective judgment, you want to be using quantitative analysis, and to do that with that experiment we can use a colorimeter instead of the human eye. It gives us a number rather than a subjective opinion. It's like, oh, that's a bit red, what do you think? No, I think it's orange. No, don't do that. Use a colorimeter and you get an accurate measurement. So you get a closer measurement to its true value. So precision, often confused with accuracy, but precision is the degree of resemblance among study results for the study to be repeated under similar circumstances. So how close are the measurements to each other? In my little revision guide, precision is saying that precise results do not vary much from the mean. Precision is reduced by random error, the unpredictable way in which all measurements vary. Okay, so, and it states that here. So precision is altered by random error. And to make something more precise, we should be thinking about the equipment we're using, the units it's got. So if you've got really large units, so if you're using centimetres and not millimetres, then to get an accurate and precise measurement is harder. Okay, so you want to use smaller units, better equipment, the technique in which you're measuring something. So when you're looking at the meniscus, right, I'll just literally quickly pause the video and put in a picture of a meniscus. Okay, I know that's not much detail, but when you're looking at a pipette and you have that sort of curve at the bottom, you want to be measuring from the bottom of that meniscus so you get eye level with it, and that's a technique that can affect your... Another thing to look at is the setup of the apparatus. So when you're measuring like the effective temperature, you want to use that water bath, thermostatically controlled water bath, but you need to give it time, the substance time to adapt to the temperature. You don't want to start recording as soon as you put it in the water bath. You want it to get to the right temperature first. So let's get rid of that now. Another example of that is when you're looking at light intensity, so the effect of light intensity and photosynthesis, you need to give the plant that you're testing time to adapt to the different light intensities or the different light wavelengths you're using. So precision, give things time to adapt to the conditions you're testing. Make sure you've got good technique for measuring it. Make sure you're using the best equipment possible and the smallest units possible to minimise errors, reduce the variety from the mean. And if you've got large error bars, this is usually indicating that your measurement is not that precise or it's not that accurate, and the use of statistical tests can help show how precise or accurate our measurements are. Right, let's now look at validity and reliability. So validity and reliability are much more likely to come up. There's a lot more to speak about regarding them. Valid results answer the original question. To get valid results, you need to control all the variables to make sure you're only testing the thing you want to. So when you're looking at validity questions, we're looking at variables and making sure you've got all of them controlled. So specifically, validity is ensuring you've got the right controls. You need to be, and by the way, I've done a past paper question on this, and validity is probably the most accurate to where you're going to get your marks. All these things were found in the mark scheme apart from this, okay? So let's speak through each one. So once again, repeats 3 times minimum. So you use repeats to identify and discount anomalies. That is a huge thing. Always going to get your marks when you're talking about identifying, discounting anomalies. It allows for statistical tests, which is a mark often people will forget about. So statistical tests often come up in this topic, and you usually get a mark if you're pretty clued. A little bit of advice. Often these questions, the ones you don't want to see come up, but they inevitably will do. And if you do not know anything on how to answer the question, if it's something like this, always mention repeats 3 times. Always mention how you can identify and discount anomalies. Those 2 things will nearly undoubtedly get you marks in these sort of questions. Then even if you get 2 out of 6, that's better than naught. Another thing to increase validity, you should use more intermediate values. So this can help identify the trend more clearly, and just learn that, because both those things were in a mark scheme. By the way, you have the point and then have the reason why. All of these in a mark scheme, so try and use them. So use more intermediate values to identify the trend more clearly. So when we're talking about intermediate values, basically make more measurements more often, because so if you're recording, let's go back to the effect of temperature on enzyme rate of reaction, you should use lots of different temperatures, so not just 20, 40, 60. Try and get intermediate values, so every 5 or 10 degrees you measure, and then it's easier to identify a trend. Okay, so the more values record, the more valid your data will be, and it also helps to identify your anomalies again. Once again, with controls, validity is all about controls and making sure everything's kept the same, and your scientific explanation will be different depending on the thing you're measuring. So sometimes you might keep the mass of the thing you're measuring the same as mass. If you use different masses, they're going to have different surface areas, and a different surface area will lead to different rates of diffusion osmosis, and that's something that will come up often. So keep the mass the same, keep the surface area the same, keep the volume the same. do not keep the weight the same. Scientists like to say mass, not weight. And the reason we try and keep things as valid as possible is because we want to avoid bias and avoid systematic error. And this point again, to identify and remove anomalies. Sorry for me repeating myself loads, but I'm just trying to drill it into my head and yours. Finally, we've got repeatability. So how repeatable is the experiment? You should be getting the same results each time if your experiment is very reliable. Will one get the same values if the measurements are repeated? So once again, 3 repeats minimum. You should be using means. Okay, so you repeat things and you find out the mean. You need to be considering all your variables, dependent, independent, and control. How easy is it to control these controls? How easy is it to measure the dependent variable? How easy is it to change your independent variable? So we want to be getting the same results every time. And to measure reliability, once again, similar to precision and accuracy, you want to be looking at error bars, statistical tests. The smaller the error bars, the closer, the more reliable the data is. If you've got really large error bars that are crossing over, that will tend to mean it's unreliable data. Okay, so quite often you will get a question asking you the limitations of this experiment. So it will give you this experiment that a student did. It will ask you what's wrong with his method or what's wrong with this experiment, and you should speak about all these things. So vroom car, the analogy I made, I'm going to copyright it. No, I'm only joking. Anyone can use it. I hope you like it. Anyway, let's speak through each one. V is for validity. So how valid are his results? And remember, we're going to be looking at how he uses his controls, the variables, dependent, how did he measure it, independent, what did he or she change, and how did they change it, control, how was it controlled, did they have a control, did they have a negative control, okay? If they didn't have these things, mention it, say that they were weak in these areas. Something else here is independent variable. How do they change it? Did they, if we're looking at concentrations, to get different values of different concentrations, did they use a serial dilution or did they just guess the amounts? Okay, look at things like that. So validity is V, R is reliability, and I've spoken to you about both of those before. O is observations. How did the individual make his observations? Okay, his or her observations. Did they use objective measurements, which is the next one? So remember, like quantitative measurements are objective measurements. They're not dependent on opinion. That would be a good thing. If the individual's using subjective measurements, that's not so good thing. And I've highlighted that yellow because that's sort of something I've added in myself to make the analogy work. But it does make sense, and it could come up in a mark scheme. Maybe not in that wording though. So when you're talking about objective measurements, that's just a way to measure, just a way to remember it in this analogy. I'd probably be speaking about if his method of measuring, how he observed it or she observed it, if that was valid, if it was if it involved human error. So like, did they measure it at eye level? Sometimes it won't be specific, it will be really vague, and you just have to assume these things and just mention them. It might say he measured the line, he measured the amount in the pipette, and it doesn't tell you how he measured it. So did he get? So you should just assume that they did, and mention these things. Should get eye level. Should measure from the bottom of the meniscus. Okay, so M is for monitoring, and that's related to reliability. How did the student monitor their results. So then we've got controls, and controls, as we've already spoken about in our variables, did they have a control? Did they have a negative control? A is for anomalies. Remember, identify anomalies and discount them. Have they identified anomalies in their experiment? Sometimes it will give you a table, say this is their table of results, and you see that there's anomalies in it and they should have discounted them, but they didn't, so you speak about that. And R is the reasons for the above. So don't just mention all of these. Don't just say he was not valid, it was not reliable, his observations weren't good, he didn't use control, he didn't identify anomalies. Explain each point. R is reasons for all of the above. So don't just be really vague and just mention each one. Really be thorough. And when I'm looking at this question, say it's a 5 or 6 marker, I think you're definitely going to be hitting marks for validity, reliability. You'll hit a mark for each variable you mention. Talking about these observations, I'm pretty sure you're going to be hitting a mark when you're looking at that. If the way they made observations, if their method was invalid, okay. Anomalies, if they even repeated the experiment, made a mean to identify and discount these anomalies. If they didn't do that, you need to be mentioning that, and that should be getting you marks. Obviously I can't guarantee this, it's the first year this exam's ever been, we've not really got past papers to look at. Anyway, that's where I'm going to leave that limitations of the experiment question with a vroom car is your way to remember it. Validity, reliability, observations, objective measurements, monitoring, controls, anomalies, and reasons for all of the above. Okay, so finally let's look at how to design an experiment. So first of all, so a question might ask you, it might tell you, design an experiment to measure blah blah blah. I can't think of a good example. I'll look at one later. So you need to design an experiment. So what are you going to do? First of all, you must state all 3 variables, what they're going to be, how you're going to measure them. So dependent, independent, and control. Mention all those 3. I'm pretty sure you're going to get 3 marks already. Okay, as long as you give enough detail about each one. Then there's going to be marks for saying how you're going to make it reliable, valid, accurate, precise. So basically just stating how you're going to measure your variables, how to measure, how you're controlling it, the units you're using for measuring, how often you're going to measure. If you're going to repeat it 3 times, which you will state, I hope everyone who watches this video will state, repeat the experiment a minimum of 3 times, not just repeat it. That will get you the mark. The use of means, and using these means to remove anomalies, using error bars and statistical tests, okay? All these things are big marks. How to control your variable, your use of a negative control. Literally just stating something as simple as use distilled water as a negative control, that will get you a mark. Your scientific method, so while it's asking you to design a method, design an experiment, there'll only probably be 1 or 2 marks maybe for the actual method you use, how you're doing it. So be really brief on that, but focus on these other areas because these are where the marks are at. You will nearly always get a mark for your safety precautions, so stuff like wearing goggles, wearing gloves, if you're using toxic things, use a fume cupboard, things like that, all things you should be aware of. Just make sure you know a list of safety precautions and pick the one that's relevant to the experiment, and you should get a mark for safety. Discuss sample size, so you want to use as large a sample as possible to make your results more valid and accurate when you more valid and reliable when you make these means. And the final thing is removing anomalies, which I've already spoken about. So let's give an example of a question I made up. So you want to, that it asks you to design an experiment to measure the effects of light intensity on photosynthesis. Usually it will not be something, a topic you've ever met before, and you have to really use your head a bit, but most people know this experiment. But first of all, dependent variable. So we want to measure this. I'm going to measure the amount of oxygen released, because I know oxygen's a byproduct of photosynthesis. So how can I do this? I could count bubbles, but I might not write that in the exam. I might say I could use a gas syringe, measuring it in centimetres cubed. That's given my units, and using a gas syringe I can make it as clear, as valid as possible, as precise as possible. Counting bubbles is not as accurate. Okay. Independent variable. So our independent variable is our light intensity. So how am I going to do that? I'm going to move a lamp different distances away from my plant. Okay, then we've got control, and you're going to list a bunch of different controls. You might only get 1 or 2 marks for this, but list as many as you can. So I'm going to keep the same: CO2, temperature, the type of plant. I'm also going to have a negative control. So when I'm measuring the effect of light intensity, I didn't actually write this down, which is bad of me, but my negative control is going to be a dark room. So I'm going to have one plant in a dark room, no light, and it will give me a negative result, and it will prove the different light intensity is having an effect. Repeat 3 times. Just by stating that, I've got a mark. And if I say repeat 3 times to then create a mean of results, and identify and remove anomalies, that potentially is 3 marks. If you're unlucky, it will be 2. 3 things you state there: repeat the experiment 3 times, create a mean of results, and use these mean of results to identify and remove anomalies. 3 marks in a 6-mark question. Then you'd state your other 3 variables, and then you state a safety precaution. I was struggling for this. I just put wear goggles. I don't know how, if that would even get me a mark. Maybe try and think of something yourself. Comment it down there if you've got a better safety precaution for this experiment. But look, the point I'm making is I've listed 3 variables, how I'm measuring them, and I've also put repeat 3 times, create a mean of results, and identify and remove anomalies. And I'm pretty sure if you mention those 3 things, these 3 variables, and 1 safety precaution, you could be looking at least 5 or 6 out of 6. If it's an 8 marker, maybe you need to be looking this side to add a few more things. So your method, because look, I've mentioned all these things. I've not really actually mentioned my method, my apparatus, my sample size. Okay, there's so much to talk about. Don't waffle on about each individual thing. Just get your points down and get those marks. That is the end of this video on Unit 1.1. Thanks for the suggestion. Sorry, I can't remember who suggested it, but good luck in the exam on Monday if you're taking it on Monday. Probably won't put any more videos out before then. Thanks for watching.